Hi guys, it's me, and since I'm not seeing you in class, I thought I'd give you a screencast to finish up the notes for this part of the unit, and you'll have some other work that you'll need to do to just catch up. So I left off talking about the different prokaryotic versus eukaryotic cells, and there's a spot in your notes where it asked you to sort of do a Venn diagram comparing them. And I just wanted to review that a little bit. Remember, prokaryotic, pro means before nucleus and eukaryotic means after. So prokaryotic cells are really simple. They don't have a nucleus, though they do have this nucleoid, which is like a tangle of circular DNA that's floating freely in the cell. They have no membrane-bound organelles. They're much smaller. They divide through a simple process called binary fission that we'll talk about next part of the unit. They're mostly unicellular, and they can be both anaerobic and aerobic. Eukaryotic cells are much more complex. They have membrane-bound organelles. They're typically a lot larger. They're usually a part of a multicellular organism, and most of them are aerobic. And then they both have commonalities, like both have cytoplasm, both have a plasma membrane. Some of them can have cell walls and vacuoles. The ribosomes are in both, but they're slightly different, and they both have genetic material. So if you want to take a second to write these down, you can. But the big question is, why aren't cells any bigger than they are? If you look at this diagram here, the biggest cell is a frog egg cell, and it's like 1 millimeter, but the rest are much, much smaller. So what's keeping them in the 5 to 50 micrometer in diameter range? Well, we know that cells need to communicate, and we know that they take vital material in and get rid of waste out of their cell membrane. So the problem is, if cells get too big, their surface area isn't going to be enough to meet the needs of the volume of the cell. So they won't be able to take in enough nutrients and waste. So if you look at a typical unicellular organism, an amoeba, which actually has a nucleus, it's a protist, it's eukaryotic, it needs to take in oxygen through its cell membrane, and it needs to get rid of carbon dioxide. And if it got much bigger, the surface area would grow, but it grows at a cubic rate, and the volume grows at the surface area grows at a square rate, and the volume grows at a cubic rate. So larger cells would have to import more material through the cell membrane, but it would also have to meet the needs of a larger volume. So if we looked at Shaquille O'Neal, who all of you guys probably know was a basketball player who was 7 feet tall and 300 pounds. If he were double the size, would he be double as good of a player? Well, the same surface area to volume principle illustrated by cells exists with him. If his height were doubled, his proportions would be the same, but his surface area would be quadrupled and his volume would be octupled, which means he would weigh like 2.5 tons. Not only would he not be able to rebound anymore, but he probably would be crushed under his own weight, sort of like a blue whale. He'd have to live in the ocean from then on. So cells don't get huge, and part of that is the constraints of this surface area to volume ratio. And if they get really big, they're hard to maintain. We can see this with our lungs. Our lungs really maximize surface area to volume ratio. Instead of just being a big balloon on either side of your trachea, they actually have these branches, and at the end of each branch is another branch, and at the end of each of those little branches are all these little circular vessels called alveoli, which allow you to exchange a ton of gases. So it's maximizing the surface area to volume by making branches and branches and branches within branches with little circles at the end, instead of trying to just exchange gas with that big sac right there. Animals utilize this surface area to volume ratio. Animals that live in warm climates, like the desert hare and the elephant, have certain features that allow them to have a lot of surface area to cool their bodies. Look at these long, lanky legs and ears. Look at these big, flappy, thin ears. They have a lot of surface area, and they can cool that way. Whereas these two who live in the Arctic, they don't want to have a lot of surface area to lose heat, because they want to stay warm, so they tend to be stockier. What I want you guys to do for homework is read this Agar Lab, and we'll be doing this in class on Wednesday, but we're pretty tight on time. We're going to be using agar, which is this like gelatinous material made of seaweed, and we're going to make 1, 2, and 3 centimeter cubes, and then you're going to put them in a dye and see what happens in terms of diffusion. So if you guys could write the hypothesis before you start, that would be great. So before next class. Not all cells look the same. Here's 4 very different cells. Nerve cells, which are down here, have really long, skinny tendrils so they can transmit messages. Red blood cells, really circular to maximize the surface area to volume so they can exchange gases. Skin fits really tightly together. It's our protective barrier, so it's important that those cells are packed tightly. And sperm cells are tiny, and they actually are super streamlined. They have no organelles except for really the DNA in the chromosomes. It's because they want to make it really far and really fast. But how did eukaryotic cells come to be? Well, there's something called the endosymbiotic hypothesis. Remember endocytosis, where one cell engulfs a particle? Well, in this case, one prokaryotic cell, the purple one, engulfed a green prokaryotic cell, and instead of digesting it and eating it for lunch, it allowed it to live inside of its body symbiotically. And it became what's known as probably the organelles that we know of as mitochondria and chloroplasts. And we know this because they both have double membranes, they both have their own DNA, which is similar to bacterial, and they also divide on their own through simple diffusion. So sometimes we call this the Pac-Man analogy, but Pac-Man didn't digest the small prokaryotic cell. He just let it live inside and become an organelle. And that's how eukaryotic cells came to be. One of the things you guys are responsible for learning are the organelles of the cell, and you'll be teaching each other about the organelle that you researched on Friday. But I'm also going to ask that you do a little work on this something called the Cell Analogy Project. One of the sites that'll be helpful, is this Inside the Cell website, where you can kind of look inside. You can turn this into a plant cell, and then you can look inside and click on each of the different organelles, and it'll tell you about chloroplasts, vacuoles, and it'll differentiate between animal cells and plant cells. And I'd like you to try to figure out what's different about those and fill out the Venn diagram in your notes. But also what I was planning to do today was the Cell Analogy Project, where your mission is going to be to come up with your own analogy to help you learn the functions of all the cell parts or organelles. So if you click on this link, which I'll put in Schoology, you are going to get in a group of a pair or a group of 3 and review the organelles. I have some resumes here and a Google presentation. And I want you to fill out this chart and explain the basic function of each of these and what kind of cells have that organelle. And then I want you to come up with an analogy for something that is like that. In this case, there's a factory, so that you can see the nucleus is kind of like the boss's office. And here's a video just kind of describing the difference. I'm going to actually stop this because the sound is horrible, but I'll put that link on today so you can see an example. But that's using the cell as a city. Here's another model where they're using a cell analogy. A cell is a restaurant, and you can kind of see that, you know, like the cell membrane is sort of like the restaurant doors, and the cytoplasm is like the restaurant floor, and the cell nucleus is like the restaurant manager. So what I want you and your partner or alone to do is to figure out an analogy that works. So here's one that's a boat, and you can see they've labeled it like, oh, look, there's solar panels. Those are like the chloroplasts, and the nucleus is like the captain's quarters. So the final product is really just this table right here that I showed you, and then some sort of graphic representation of that analogy. And again, it does not have to be super refined. It can be kind of a sketch, and it doesn't have to be really professional by any means. But that will be due the following week, like sometime next week. And then one thing I wanted you to notice is the difference between plant and animal cells. You can see here that there are certain parts that they all have the same, but you can see over with the typical animal cell, they have centrioles and lysosomes, which plants don't have, and plants have cell wall and vacuole and chloroplasts, which the typical animal cell doesn't have. So plants tend to be larger. Plants like to be in hypotonic solutions to maintain turgor pressure. Plants can't change their shape. Plants contain a large vacuole, much bigger than these guys will sometimes have vesicles. And plants have plastids like chloroplasts and leucoplasts. All right, that's it for today. Hope you guys are well. I miss you.